Here we have a sample problem. Mutations in the MSTN gene are associated with abnormal muscle development in whippets, dogs that were developed for racing. Whippets with 2 copies of the mutated MSTN allele are more muscular than normal and known as bullies. Whippets with 2 unmutated alleles have normal muscle development. Whippets with 1 unmutated and 1 mutated allele are more muscular than normal whippets, but not as muscular as bullies. The MSTN gene is located on chromosome 37. Then we're given citation information. Question asks us, the mode of inheritance for the MSTN gene that controls muscle development is an example of what? We have A, multiple alleles, B, sex-linked condition, C, incomplete dominance, and D, an autosomal recessive condition. So the key here is that the heterozygote in the whippets, the ones that have the single mutated and the single unmutated allele, are more muscular than normal, but not as muscular as those who have both mutant alleles. That would be an example of incomplete dominance. Because you have blending of the 2 phenotypes together, they are more muscular, but is not quite as muscular as the homozygotes. We know that it can't be multiple alleles because we're only given 1 allele to talk about here. We also see it's not an autosomal recessive condition because, again, there is that blending of the complete dominance. We haven't talked about sex-linked genes yet, but we are told here that this is on chromosome 37. Dogs are mammals, so they have XY chromosomes for sex determination. So we know it's not on the sex chromosome, which is the condition for it to be sex-linked. We'll talk later about what exactly that pattern of inheritance looks like in more detail, but we know from this information that it is autosomal and there is no reference to different inheritance in males and females, which would be a characteristic of a sex-linked trait.